video, we are going to understand the principle behind pyrosequencing for DNA molecules. As we know, DNA bases are made up of purines and pyrimidines. Adenine and guanine are the purines, whereas thymine and cytosine are the pyrimidines. The purines and pyrimidines pair with each other. Adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. Let us consider this particular DNA strand. We know that DNA polymerase synthesizes the daughter strand in the 5' to 3' direction. This addition occurs according to the ATGC base pairing. Normally this addition is uninterrupted as all the 4 nucleotides, that is A, T, G and C, are always available for the DNA polymerase. However, in pyrosequencing, we provide each of the 4 nucleotides one by one to the DNA polymerase and see which nucleotide is being accepted and which one is not being accepted. Let us assume that we have designed a pyrosequencer in such a way that our machine will be adding A first and then D, then G and then finally C in a sequential manner, and this cycle will be repeated again and again, and we want to see that this DNA polymerase is going to accept which of the 4 nucleotides that we are going to provide. In this example, when you first add adenine to the growing strand, DNA polymerase will not accept adenine. Well, right now since we already know the sequence, we know why DNA polymerase is not accepting adenine. It is because the next nucleotide is cytosine and we know cytosine pairs with guanine. Thus, adenine is not accepted by DNA polymerase and it is degraded by the enzyme apirase. Next, thymine is provided to the DNA polymerase and again thymine is not accepted and it is destroyed by the enzyme apirase. Next, the pyrosequencer provides the DNA polymerase with guanine, and since on the parental strand cytosine is there, guanine is accepted by the DNA polymerase. During the incorporation of guanine at the 3' end, pyrophosphate is being released. This pyrophosphate reacts with adenosine 5-phosphosulfate in the presence of ATP sulfurylase to give ATP. This ATP reacts with luciferin in the presence of enzyme luciferase. Luciferin is converted to oxyluciferin and a photon of light is being generated. This signal is recorded and the output graphs looks like this. On the x-axis we can see the nucleotides that are being added and the y-axis shows the light signal. Initially we provided A and T to the DNA polymerase and they were not accepted. Thus there was no light signal, which is why you see a straight line above adenine and thymine. However, on addition of guanine, light signal was received, which is shown as a peak. This tells us that on the template strand C was present and on the daughter strand G was being incorporated. Now, once the machine records a signal, it degrades the unused guanine and ATP using the apirase enzyme and it starts the sequence of ATGC addition again. In the next cycle, on addition of A, no signal is received as the next nucleotide is A, which will pair with thymine and not with adenine. Thus, adenine is destroyed by enzyme apyrase and thymine is added. On addition of thymine, the graph shows a peak higher than the one obtained earlier. This shows that the DNA polymerase added 2 thymine molecules. Because 2 thymine molecules were added, 2 ATP were generated, and these 2 ATP could react with luciferin in the presence of luciferase to give 2 photons of light. Thus, the signal that is being generated is double. Thus, the double peak represents the presence of the same nucleotide twice. Similarly, the entire strand is extended and based on the graph, the strand sequence is discovered. No peak means that the nucleotide is absent. One peak that means that one nucleotide is present in the sequence, that is G, and 2 peaks means that 2 nucleotides are present. Similarly, if we have a longer peak than this, then we can see the intensity, and according to that we can say whether there is a presence of 3, 4 or 5 repeated nucleotide sequence of the same nucleotides. A pyrosequencer requires the DNA to be sequenced and DNA polymerase for elongation of the complementary strand. Apart from this, it requires adenosine 5' phosphosulfate and ATP sulfurylase for the generation of ATP from the pyrophosphate, which will be released during nucleotide incorporation. After the generation of ATP, it requires the enzyme luciferase, luciferin and apyrase for light generation, which will lead to signal production, and for the degradation of unused nucleotides and ATP.
If this video was helpful, then please like this video, share it with your friends, subscribe to my channel, and don't forget to leave us your valuable comments. Thank you.